to introduce our first speaker. So you see the agenda there. We're very excited about the topics that we have. I think that we really cover a range of GI. And the first topic that we wanted to start with is the microbiome of the gut. And I'd like to introduce Najwa El-Nachef. She is an associate clinical professor of gastroenterology at UCSF. And perhaps, close to my own work, she is the program director of the GI fellowship at UCSF. Hard to say which one is more work, honestly. And then interestingly and relevantly, she's the co-director of the Microbiome program at UCSF and established that program in 2014. I was interested to read how many FMTs, fecal microbiota transplants, have been done. Over 200 for recurrent C. diff and over 150 for IBD, often as part of clinical trials. So a lot going on, and a lot of expertise coming from Dr. El-Nachef this morning. She's given a number of talks on this topic. We gave her a fair amount of freedom with what she talks about this morning. And I, for one, am particularly excited to have her be our first speaker today. So Dr. El-Nachef, thank you.
Thank you for that lovely introduction. I'm very excited to give my first talk in person since the pandemic started and not into the void of Zoom. All of you on Zoom, I'm still happy to talk to you, but I'm glad there are some faces to see as well. So yeah, I'm going to talk about the microbiome of the gut. And the gut microbiome is a topic that has been really rapidly increasing in interest over the last decade. So when I look at my PubMed search, I can see that over the last decade, the amount of publications in this area have really rapidly increased. Part of the reason that there's been such a dramatic increase is that we've moved away from the culture-based method of isolating strains from a sample, and we've now found DNA-based techniques to recognize the vast array of microbes that reside in a sample. So a couple decades ago, ecologists were recognizing that when they looked under microscopy, there was much more diversity of bacteria seen under the microscope than what they were able to culture. So now we have the tools to better understand how complex the microbiome is. So there's just so much to learn now. So, the gut microbiome is a really huge topic. You could pick any little niche topic and spend far more than 20 minutes discussing it. So today I thought we'd go over some of the definitions that are used in microbiome research and talk about what we know so far about the development and characteristics of the microbiome. Of course, all of this is more interesting when we talk about the potential clinical applications, because that's what we want to do. We want to take this information and use it to help our patients. While there's a wide variety of clinical applications, I thought for this group, it would be best to focus on irritable bowel syndrome and inflammatory bowel disease. And of course, I think we do have to talk about what are the limitations to microbiome testing, because it's a very popular topic, but sometimes we kind of have to be more measured with our patients when we talk about the utility of these tests. So just going over some of the definitions, the terms microbiota and microbiome are often used interchangeably. But when we talk about microbiota, that's like the organisms that's in a sample, whereas the microbiome is the catalog of their the microbes and their genes. So there are over 100 trillion bacteria in our microbiome, and we also have fungi, protozoa, and viruses, but bacteria make up the majority of the microbiome. It's said that the microbiome may weigh as much as 5 pounds, which is sort of gross, but definitely interesting. And the human microbiome includes a huge amount of genetic information. So there's 100-fold more genes in the microbiome than in the human genome. So a lot of genetic material, a lot of potential implications from these genes. The metabolome refers to the compounds that are created by the bacteria, and this is how they confer their functions within our body. And then just a couple other terms I wanted to bring out is that we often, when we look at the medical literature pertaining to the microbiome and gut health, we see measures of diversity. So the term alpha diversity refers to the richness and the evenness of a sample. So in general, we want there to be a lot of different groups that can survive within a certain sample, and that there should be evenness. There shouldn't be just one dominant bacteria. So one example that I like to illustrate when we talk about alpha diversity and how that's associated with health, this is from the landmark study from 2013 of fecal transplant in recurrent C. diff. And you can see here that often alpha diversity is represented in a box plot. And you can see here the donors have a very high alpha diversity. Those are the people who have like a healthy microbiome. In the second column, you have the patients who have recurrent C. diff prior to treatment with fecal transplant. And you see the diversity is much lower. And then that diversity is restored with the fecal transplant. So greater alpha diversity is usually associated with greater health. Beta diversity is when you are comparing the two different communities. So if you want to know like how similar a healthy donor is to someone with ulcerative colitis, you would look at it on a PCOA plot. Each dot would represent the microbiome analysis, and the closer two dots are together, the more similar they are. So let me see if I can get this mouse here. Is it moving? Yeah. So this is a PCOA plot from a study that we did on ulcerative colitis and FMT, and the dots in the purple represent some healthy donors. The dots on the far right that are in yellow are people who have received antibiotics, and the ones here in the middle are patients with ulcerative colitis. So you can see the purple ones all kind of cluster close together, kind of showing that the healthy ones are similar. Not surprisingly, the ones on the far right with the antibiotics are very different. So just some terms that I think are helpful to know as we get into this. So we know that the microbiome of the gut starts to develop right away, right? So the mode of delivery can actually influence the infant microbiome. And in the first few years of life, it can change dramatically, especially as solid foods are being introduced. As the diet starts to resemble that of an adult, you start to see the functional capacity increase in the gut microbiome, particularly pertaining to vitamin biosynthesis and polysaccharide digestion. And then once you get to adulthood, the microbiome tends to remain stable and then becomes a little less diverse in the elderly years. So the million-dollar question is, what constitutes a healthy gut microbiome? I wish I knew. I think everyone is searching for this. There's no standard definition, but there are some characteristics that are considered to be important in defining a healthy microbiome, which include high levels of diversity, stability, resistance to stress-related change, and a high level of redundancy of the metabolic pathways. So this is a figure from the Human Microbiome Project, where they combine all the taxonomic profiles of some healthy volunteers who submitted a sample. And you can see here that there are that the healthy microbiome tends to be dominated by the firmicutes here in the green, the bacteroides in blue, and proteobacteria in yellow. So there is some general stability and healthy controls and consistency in these phyla, but at the level of the species and subspecies, there's much more variation. So it's considered like that everybody's microbiome is unique if you take it down to that level. But despite these differences, there tends to be pretty constant and consistent functional capacity when it comes to a healthy microbiome. So this is another one of those PCOA plots. This is just demonstrating that the microbiome, while you can change it and manipulate it, it tends to be pretty stable. This is from a study that's looking at the effect of diet on the microbiome. So the open circles are people who are on a plant, or when a participant is on a plant-based diet. The closed circles are samples that are obtained on an animal-based diet, and each different color represents an individual. And so what's interesting here is even though there's change with change in diet, each individual tends to kind of have a pretty stable microbiome. So here in the red, that's one person. In that we have a blue, so they all kind of cluster together, that they stay pretty similar. So that kind of goes back to how it's like harder to manipulate it because it is pretty stable. And the reason why we're so interested in whether or not we can manipulate the gut microbiome is because there are many functions that the microbiota perform. They're involved in immunity, they're involved in biosynthesis of vitamins, hormones, and neurotransmitters, as well as in metabolism. So there's a wide variety of disease indications that could potentially be manipulated by manipulation of the microbiome. So one key question is whether or not we are able to change the microbiome. Then in order to better answer this question, we have to understand, well, what affects the microbiome in the first place. As I mentioned before, the mode of delivery at birth can affect the microbiome. When an infant is born vaginally, they are immediately colonized with vaginal flora. And when they're born by C-section, they're colonized by skin flora. There are genetic features and the immune response within a human that can affect the flora. Of course, diet, antibiotics, infections, these are all things that can change the microbiome. And my favorite way to change the microbiome is fecal transplant. We'll get into that a little bit more. So what are the potential clinical applications? So when we talk about all the functions of the microbiome, we really start to recognize that there are a wide variety of effects on the host, including effects on immunity, on epithelial barrier function, on visceral hypersensitivity, motility, and what I find very fascinating as well, the interaction of the brain-gut communications. So there's a lot of potential ways that you can ways that the microbiome can affect the host. Now, we can't get into every single indication, but I think most of us here have dealt with patients with irritable bowel syndrome and with inflammatory bowel disease. So I wanted to focus on those two potential indications. In IBS, we know that patients tend to have different changes in their microbiome when compared to healthy controls. It also kind of makes sense that the microbiome is a reasonable target for these patients. We've known for a long time there's like the entity of post-infectious irritable bowel syndrome, so it would make sense that there's some sort of alteration in the microbiome in that setting. We also know that there's a subset of patients that respond to rifaximin and other antibiotics. So this is, like, of course, an area that is pretty intuitive to investigate. So no patients with IBS may have a lower abundance of butyrate-producing bacteria. This can affect intestinal permeability and nociceptive sensory pathways. They also can have alterations in methane production. That can be lower in patients with IBS-D and higher in patients with IBS-C. They also can have an increase of abundance of proteobacteria and firmicutes and a decrease in bifidobacterium and fecalibacterium. But the clinical significance of that is still not very clear. And then in inflammatory bowel disease, the gut microbiome is definitely perturbed. It makes sense because because the healthy bacteria tend to thrive in an anaerobic environment, and when there is inflammation, it's more of an aerobic environment. So if the environment isn't good to begin with for healthy bacteria, it makes sense that there would be a difference. Patients with inflammatory bowel disease are known to have decreased diversity. That is one of the clear things, consistent things we see in patients with IBD. They also have a lower amount of bacteria that are associated with health and the production of short-chain fatty acids, such as Rosabura hominis and Clostridial 4 and 14 species, and they can have an increase in their profile of some bacteria associated with inflammation. And while we don't know if it's the chicken or the egg, when we look at the interaction of the gut microbiota with the epithelial barrier, we do know that there is an interaction with the bacteria that can result in an increased hyperactivation of Th1 helper cells and a decrease in T regulatory cells, which results in chronic inflammation. So what do we do about this, right? We have some information. It seems intuitive that maybe we can try to manipulate the microbiome and see if this results in a better outcome for our patients and change the pathways that are leading to symptoms and or inflammation. So probiotics, I think if I asked for people to raise their hand for how many times their patients, who've had patients to ask about probiotics, I think it would be 100% of us would probably raise our hands that people are really interested in probiotics. In 2015, there was a study that estimated that 3.9 million Americans were taking probiotics, which was up fourfold from just 2007. I imagine that number is probably even higher now. But we don't have a lot of data to necessarily recommend probiotics for our patients. So last year, the AGA did a technical review on probiotics for various GI indications, but none of and this of course is a busy slide. I just put it up here for you guys to look at the last two columns where you can see the strength of the recommendation and the quality of evidence overall is pretty underwhelming. For most indications, though, there was either a knowledge gap or low quality of evidence. And the only two indications that had a moderate degree of evidence were for children. So for the prevention of necrotizing enterocolitis in preterm infants and for acute infectious gastroenteritis in children, there was a potential for probiotics to be helpful. All the other indications that were looked at in this review did not seem to be very promising, based on the positive data, not necessarily that there were a lot of studies that were showing like a negative result. So for IBS, the quality of data was low. It was not recommended outside a clinical trial. For Crohn's disease, for ulcerative colitis, similarly, we did not see a big benefit. Pouchitis, interestingly, does have, when we think about IBD, that's the one with the most data. Even that only had a very low quality of evidence. So still, we need a lot more information. What was also interesting is that a couple years ago, there was a study that was published in Cell that indicated that that probiotics may actually perturb the gut microbiome. So this is this kind of blew a lot of people's minds. This was a small study where they looked at both a subset of mouse models and also healthy human volunteers and gave them antibiotics and watched what would be the best way for their microbiome to be restored after the disruption from antibiotics. So this one, of course, I'm only pulling up the human one for us, but you can see that at the control samples had high diversity. Not surprisingly, with antibiotic administration, the diversity was very low. The microbiome was restored most quickly when autologous fecal transplant was given. And then in the red, we have the spontaneous recovery, so like no therapy was given afterwards. That was actually better than the group that received probiotics. So kind of surprising, not that intuitive. We're still trying to understand what's going on, but I think this is just an example of how just because it's sold as being a good beneficial bacteria, we really need to see the data before we recommend it widely. Next, another way that we can consider we can manipulate the microbiome is through fecal microbiota transplant. Neil Stolman is here. He is the one who actually taught me how to do this. So I'm really thrilled to be able to show that. I think one of these pictures may actually be yours. So we already know that fecal transplant works pretty well for C. diff, so I won't really get into that today. But the question is whether or not it's effective for other indications. In IBS, the data has been pretty mixed. This is a meta-analysis that was published a couple years ago that showed the results of 4 randomized controlled trials. There was a lot of heterogeneity in these trials, but overall the message is that maybe not effective. I think we just need to better define the patient populations and do more studies before we can make a determination whether or not it's helpful for IBS. When it comes to IBD, for fecal transplant and Crohn's disease, really there's not a lot of great data. There's no large randomized controlled trials that have been published. We had a trial at UCSF that we actually halted early because we had a couple patients that flared after FMT. However, FMT for ulcerative colitis does seem a little bit more promising. I could spend far more than 20 minutes talking about all these studies. There's some really cool lessons from each one of these studies, whether or not there's a super donor, whether or not we should be doing repeated fecal transplant, whether or not we should be anaerobically preparing the stool. So if any of you during the break want to talk about this in more detail, let me know. But what I do want to, the take home from this is that there have been three out of the 4 randomized controlled trials have shown that fecal transplant has a statistically significant improvement over placebo for both clinical and endoscopic remission. So I think that's pretty promising, and that's what we've been studying also at UCSF. So promising for UC, not so much for Crohn's. There are multiple other methods to manipulate the microbiome, of course, although, rifaximin alone deserves its own topic, but we can leave that for another time. Prebiotics are also of interest, although we don't have a lot of data for that yet. That's giving the substrate that's like within the food, the substrate that will help promote the growth of healthy bacteria. And symbiotics would be a combination of probiotics and prebiotics. And then, of course, there's a lot of interest in diet as well. But these don't really have a lot of robust data. So are we there yet? Are we able to take this result that your patient brings to you and decide how to change the management of the patient? I don't know, but by show of hands, how many of you have had your patient bring one of these to you and ask you what to do? A decent amount, right? So, you have a patient, they'll come to you and they'll say, I have a higher ratio of firmicutes to Bacteroides than expected. I always tell them, unfortunately, we can't really make any decisions based on this kind of testing. There's quite a few limitations to microbiome testing. To start, we don't know what the clear reference range is for what is normal. And the part that I like to explain to patients is that a lot of these tests actually are reporting things to the level of the genus, but each genus has a lot of species and subspecies, and the variety of strains can have different effects. So the example that I like to use is that if you have a result that says you have more E. coli than expected, well, E. coli has different strains that have vastly different functions. So E. coli Nissl 1917 is a probiotic, whereas E. coli 0157H7 can cause debilitating illness. So it's really hard to say. That's part of the problem with these tests, that we don't really get enough granular detail. And even if we did, I'm not sure how much you would change your management based on those. It's also unclear at this point in time whether or not in all these studies that we're seeing is the microbiome causative or associative with these conditions. So that's another reason why we can't really interpret these tests. It's unclear how to best manipulate the microbiome. The probiotics, we don't see great data yet for this. For FMT, it's very exciting, but there's concerns for safety. And even if you could give FMT, at this point in time, FMT is only allowed to be given for recurrent C. diff unless there has been an IND from the FDA and in the context of a clinical trial. So there's quite a few limitations to these tests. Okay, so to answer my own question, we're not there yet, but the future is bright. Sometimes I feel like when the patients are coming in to talk to me about microbiome-related things, I'm a little bit of a downer because I'm waiting for some really good data. But there are some really interesting things when it comes to the microbiome. And I'll just I won't get into too much detail, but we're seeing really cool things like manipulation of the microbiome with FMT may actually modulate response to immunotherapy. Some pretty neat stuff with melanoma and immunotherapy after FMT. The microbiome may actually serve as a predictor for response to certain things. There was a study that looked at children and showed that a certain microbiome may predict a response to a low FODMAP diet. So that could be a potentially useful tool. Of course, we know FMT is great for recurrent C. diff, and that's been a very difficult condition to treat for many years. So that's been a relief to have this kind of therapy. There are multiple studies in UC that have shown some promise. And what I think is also really fascinating is that there have been microbial cocktails that are being developed instead of whole FMT to kind of have a more refined approach to treating these conditions. And there's been some preliminary data that's promising for C. diff and UC. Also, when you look at clinicaltrials.gov to see how many studies there are on the microbiome, there are over 2,000 studies and over 400 studies looking at FMT for a variety of indications, which just says to me that a lot of our questions hopefully will be answered. In either way, it may support or deny the use, but I think we're going to have more answers soon. So I do feel the future is bright. So in summary, the gut microbiome is determined early in life, but can be manipulated. IBD and IBS are associated with altered microbiomes, and there's some promise that manipulating them may alter the clinical course, but it's not quite ready for prime time. Probiotics and FMT are generally safe, but we do need more data to determine the proper indications and the best preparations. And finally, the gut microbiome knowledge is in its infancy, but there's great potential to use this to create novel therapeutics. So thank you, and I'm happy to take any questions.